My name's Hugh Possingham. I'm the Queensland Chief Scientist, and we're here to have a panel on remaking the coral tree of life for the World Science Festival in Brisbane. Very excited to be discussing this fascinating issue. Corals are so important to Australia. But let me begin by remembering that we are on the traditional lands of Australia's First Nation people. Here, the traditional lands where I sit of the Yagara and Turrbal people, but also recognising that they are our first scientists, our first communicators of science. And then all the traditional owners of the lands up and down the Queensland coast where the Great Barrier Reef is, and the knowledge that they have on marine stewardship that we hope in the future will be passed on to help us to save this incredible natural asset. We're going to be talking a lot about corals and coral reefs, their taxonomy, and then we'll start moving into conservation and what we can do to save them. I personally, I think, know less about coral reefs than almost anybody listening to this call, so I'm going to be asking a lot of fairly naive questions. I was involved from a mathematical perspective in the rezoning of the Great Barrier Reef in the early 2000s when we successfully brought the conservation area from just 5% up to over 30%. And I'm involved from a government perspective on many aspects of how do we manage it into the future, and have great respect for the scientists. We have the world's best coral reef scientists, there's no doubt about it, the world's best in the world, the best in the world, and it's a privilege to talk to them today. That said, what do I know actually about the details of a coral reef? I have been snorkelling a few times. I am technically known as Australia's worst swimmer, so I wandered around and flapped around. I can do it for about half an hour before I'm exhausted. And I am struck, I suppose, as like all of you have who've been out to the Great Barrier Reef, just on the diversity of that system. I don't know what I'm looking at. I know enough to know I'm seeing dozens of species of fish, most of which I can't identify. I can sort of see there's some different corals, some different structures, some stag corals, some brain corals, some fanny sort of corals. But to be honest, I don't know whether I'm looking at 15 species of coral or 2,000 species of coral. So I'm very interested to find out what we know about coral taxonomy, how corals work, and what we can do to save them. So we're very lucky to have two experts on coral reefs and coral reef taxonomy to explain this complicated ecosystem to us. Two experts from the Queensland Tropical Museum up there in Townsville. Firstly, we have Tom Bridge, and Tom is the curator of corals and also affiliated with the Coral Reef Centre of Excellence at James Cook University. And secondly, we have Peter Cowman. Peter is the curator of biosystematics. He also works at the museum and is affiliated with the James Cook University Coral Reef Centre of Excellence. I gathered that Tom is more out there trying to find the new corals, and Peter is out there looking at corals but also doing a lot of the work on the genetics, and they work together to try and untangle this diversity we have. And you know, as a person who wanders, Tom, going to you first aimlessly for half an hour, say at Heron Island or Lady Elliot Island, trying to work out what I'm looking at, I don't really know. I can tell I'm seeing half a dozen species, but I could be seeing 100 species. You know, how many species of coral are there in the Great Barrier Reef or even the world? And do we know, have we worked this all out? For, say, my field, birds, we sort of know how many bird species there are in Australia, and that hasn't changed that much.
Yeah, no, it's an excellent question. The short answer, which I think is really exciting from a scientific perspective, is that no, we really don't. We're really just starting to uncover the diversity, and as this panel will discuss, there's been some really exciting new tools and techniques developed relatively recently, just in the last few years, that are allowing us to understand these questions. But yeah, it is very confusing. Corals. A lot of the time you can have species that are the same species that look quite different because they grow in different habitats, a bit like trees. So while birds and most vertebrates don't vary that much generally in terms of their appearance within species, corals and some other invertebrate species can vary a lot. But then conversely, we're also finding that sometimes we have species that look very, very similar that are only very distantly related. So this is some of I guess that the questions that this recent research, particularly the phylogenomics, is starting to be able to resolve for us. So I certainly sympathise with you. It's very confusing. You know, we were doing some field work at Heron Island about this time last year, and I was out there collecting corals. And having studied corals for a long period of time, it doesn't necessarily get a lot easier when you're swimming around. It's still confusing.
So you're telling me that I could be looking at two things that look very similar, but they're actually not at all similar. But then I could be looking at one species looks very different in different places. That's not helpful.
No, it's not helpful at all. It is. It's very confusing, and that's been one of the issues, I guess. We've been studying corals as the scientists, and classifying them from a taxonomic perspective for almost 300 years now, going back to Linnaeus. He described the first species in the very first taxonomic work. And really, for the first 200 years, people actually never observed corals in the field. They only really observed corals, either sometimes some scientists went in the field and walked around on reef flats, or sometimes they were dredged up on scientific expeditions in the 1800s. But for the large part, most of the people that were working on corals had never actually seen one alive. They were only getting fragments of skeletons that were brought back. In the 1960s and 70s, people started scuba diving and observing them in the field. And we really started to realise that, some of these species could vary a lot. So that led to people thinking that a lot of the variation we were seeing was just due to growing in different environments. So if you grow on a reef crest where there's a lot of wave energy, you need to be quite tough and compact. If you're in deep water, you need to be able to capture light. There's not a lot of light in very deep water because it declines with depth. So in order to get enough light to survive, they need to be much more finely branched and often much flatter. So they can look quite different. But what the genetics is telling us is that sometimes we have very distantly related species that have actually evolved the same, solution to these common problems. And this is the case with corals. We see this in a lot of other groups of animals as well, known as convergent evolution, where different species come up with a very similar solution to a common problem, and therefore they look quite similar. So it's often quite difficult to tell these different species apart unless you have a very good fossil record. But fortunately, the genetics is allowing us to do that.
So by analogy to something most people know a bit more about is plants. We do aware that some plants, if you grow them, some acacias will turn into a 40-metre tree, but out near the edge of the desert, they'll be a small shrub. So we see morphology, but of course the beauty with plants is when they flower, the flowers are where you go and the seeds are where you go, and you can identify them. And so there is no equivalent, I suppose, for corals. You can't find some part of their anatomy that is resistant to changes in the environmental circumstances in which they grow?
Oh, yeah. Look, excellent question. Look, there for some groups there is. We're finding them. But again, I guess one of the problems that we had was, again, for 200 years, it was looking at specimens and saying, well, this one looks different to that one, or this one looks very different to that one. So therefore, they're likely to be different species. And so one of the advantages of the phylogenomics is that we can use this information to try and find informative characters. So you can say, well, we were classifying them using these characters, but they're actually really influenced by the environment, whereas these other characters actually seem to match the genetic data much better. So, for example, for some groups, we're finding these very, very small, what we call microstructural details of the skeleton are actually quite phylogenetically informative, which is useful, but the problem is you can't usually see these in the field. You've got to take the specimens back to the lab, examine the skeleton under an electron microscope to really be able to see these features. That makes it difficult if you're trying to do a survey in the field.
Yeah. So you're in a museum, obviously, in the Queensland Tropical Museum, part of the museum network, and you've got specimens that may be 50 or 100 years old. Are they good enough for you to be able to do what you just talked about? Some of that morphological stuff, they still good enough?
Some of them are, yeah. It depends entirely on what the question is. So for the species where we do know what those characters are that are phylogenetically informative, absolutely, we can use those. Sometimes not so much, but one of the really important things about museum collections that we're finding is the type specimens. So trying to resolve the taxonomy and the biogeography of corals, when a species is named, part of the process is you have to designate what's called a type specimen, and that specimen is the single specimen that represents the species. So, well, if you're holding a colony of the type specimen, that's the only time you can be absolutely sure that that specimen represents the species that you're talking about. And that's just not for corals, that goes for any species at all. Anything else is just a guess. So, these type specimens are really important. And what we've had to do is actually use these old type specimens and the information surrounding them in these old taxonomic works that they were published, often these beautiful big monographs, and go back to where these specimens were collected, sometimes over 200 years ago, try and find things that look exactly like that particular type specimen, and then recollect them and be able to collect tissues, in this case, that we can then use for sequencing. And this is one of the ways that we've really been able to get a better understanding of the diversity and the biogeography of corals from across the Indo-Pacific.
Yeah. So that's important for people to know, in a sense, the fact that these type specimens are in a museum and everybody knows where they are and they're grounded in that one thing, that sort of stops chaos reigning, stops somebody in another country just collecting a whole heap of corals and giving them completely new names, which would be chaos, wouldn't it?
It would be. It should stop it. It doesn't always.
That's interesting.
Look, it's getting much better. So yeah, these type specimens are really, the crown jewels of museum collections. They're really important for understanding biodiversity. Yeah. And so museums treat these type specimens very, very carefully. They're always under lock and key. Oh. And one of the things, I guess, going back a couple hundred years, it was very hard to see if they were under lock and key, say in the Natural History Museum in London, and someone was working on corals in the US, then, they didn't necessarily have access to all of these specimens and weren't able to examine them. So for that reason, sometimes these species were described multiple times. Fortunately now, one of the big things museums are doing is trying to use digitisation tools, so things like, photogrammetry and things, to make these old type specimens available. And this is actually going to make it much easier for scientists, not just taxonomists, but people trying to put names on corals in the field where they are, trying to make sure that these names are correct and they represent that actual species that they're working on. Good.
So that gives us the history and the basics of coral taxonomy. And again, interesting that such something is still not resolved. Maybe I can go now to Peter, and Peter gets into the DNA and the base pairs and about what these corals are working from the lab. And of course, we're all becoming much more familiar with DNA because I and my wife, in fact, sent off to 23andMe and we sent off some saliva samples, and that they found all our DNA. And I found I had the most boring ancestry in the entire planet. All my DNA comes from northwestern Europe. So I was extremely disappointed. I was hoping for something more interesting. But DNA can be done very quickly. Peter, can you tell us a bit about what that understanding of genetics of these organisms is telling us about their taxonomy?
So yeah, we're basically acting like Ancestry.com for corals. And it's really, really exciting because whereas in the past, like Tom said, a lot of these species were based on, these species descriptions are based on how they look. What we're finding is that there's a lot more new species than we realized, and that some of these species are not as closely related as we thought. So it's really, what we're doing is rewriting the family tree for different corals. And it's quite a task. And so if you think of a genome for any organism, like the human genome, it's nearly 3.2 billion letters long. And so when I say letters, I'm talking about the base pairs, what we actually look at when we study DNA. So the strings of repeating A's, T's, C's, and G's. And I spend a lot of time looking at it. So that's a lot of fun. It's like playing a big game of Tetris sometimes. And so if we think about a coral genome is maybe only 600 million letters long. And so that's still very long. So it's like, if you put all those letters together, it's like reading all the Harry Potter books 120 times. And so that's about equivalent to the number of letters.
That's exhausting. I only got to book number 4, let alone reading the whole series 120 times. So 600 million base pairs is a lot of data. I mean, it sounds incredible. How long does it take you to get that data, and how do you store it, and how do you even make any sense of 600 million G's, T's, A's and C's?
And yeah, so that's where it's really starting to change now. Whereas before we used to just look at small parts of the DNA. So in fact, a lot of our knowledge of species genetics today is based on what we call mitochondrial DNA. And so the mitochondrial genome is another part of DNA that we all inherit maternally. And so what has been done before is there's areas of the mitochondrial genome that have been really, really useful for identifying species, and often they're called barcodes. And so for lots of vertebrate groups like birds and mammals and fish, they've been really useful for identifying species, identifying their relationship to each other. But in corals and more widely across their relatives, anthozoans, the mitochondrial genome hasn't been very useful. They just don't have enough changes in their base code to allow us to get any meaningful information from them. But now we're starting to look beyond just single gene regions or single parts of the genome, and we're scanning thousands of areas of the genome.
So how many species or types of corals do we have the full 600 million base pairs? Do we have, have you there out there got thousands of individuals all the way down to the 600 million base pairs, or do we have them from all over the world?
It varies, and also the genome of different coral species varies in size as well. Right. And so I think if you're talking about species level, there's probably maybe 18 to 25 species level genomes that are of high quality, and we know a lot about their structure and where the genes are coding and stuff like that. And then there's more data within species where we've done things like what's called shotgun genomics, where we sequence lots of the genetic code and then try and map it to a genome that we know more information about. And so we're in our work, we're taking a different approach where we've actually used all these whole genomes that have been sequenced for different species, and we line them up and we go, okay, well, how many areas here are common across all these genomes? And we can make what's called a bait set to target those areas. But even though those areas are highly conserved, they may not tell us a lot of information. What we're pulling out is the flanking regions that have all that variation that can tell us a lot more about the ancestry of these different species.
Right. So obviously you're learning not just about the relationships between different coral species, you're also learning about the genetic diversity within a single population or species as well.
Exactly, and that's where it becomes difficult, and that's where we're trying to decide, okay, well, one person might call a population a species where it could actually be exchanging genetic data with a population close by. And I think that's where we're really we're trying to equate the 200 years of taxonomy that's been based on looking at these skeletons with what the DNA is telling us. And so there's a lot of names out there for species. Right. And so, I mean, there's one particular group that we look at, the branching corals. There's currently only about 220, or there's currently only about 120 or so accepted species. Yeah. But the 200-year history of taxonomy, there's about 400 names that have been placed on species, but then someone else decided, no, that's the same as this species, and so they merged those names together. And what we're finding is actually some of those old names are actually still important and could be valid species.
So, say, let's go into that group. The branching corals, or the Acropora group, is that what they call them?
Yeah.
Yeah. So these are
Acropora, yeah.
And they're pretty common in the Great Barrier Reef, and presumably, roughly, what would the sort of accepted diversity in the Great Barrier Reef have been, say, 10 years ago of Acropora species? Based on the morphology, ignoring all the recent work.
There's probably about 70 species, I think, in the GBR, accepted species. Yeah, and so somewhere around there.
So you're expecting that because of this work, that number could get substantially bigger, and their relationships to things in, say, the Caribbean or the Red Sea or Hawaii could substantially change.
Yeah, absolutely. So there's certainly more species within regions. So there's more, again, because of this. Staghorns are a classic, those branching staghorns. You would have seen these beautiful fields of them at Heron Island.
Yeah.
And they all look a little bit different. This almost looks like, a garden patch. There's little bits and you go, oh, is this 2 species or 1 species that's variable, or is there 12 in there? And so, this is part of the story. But I guess one of the most important things in terms of just pure diversity is that we thought that many coral species were really widespread. So occurred from the central or even the eastern Pacific, right the way across through the Great Barrier Reef in Indonesia, across the Indian Ocean to East Africa, up into the Red Sea, and as far north as Japan in the Pacific, and sometimes into Hawaii, and even across into the Eastern Pacific. So these huge ranges. But what we're finding is that, that's not the case. The species in more peripheral locations, in particular, like the Red Sea, are very distinct from the species, for example, on the Great Barrier Reef. So I guess that's a really important aspect of it. So while there is increased diversity because of things like convergence within the Great Barrier Reef, a lot of the increased diversity is because what we thought was one widespread species are clearly several different species in different parts of the world.
So this is really important for conservation, isn't it? So if coral reefs in a certain region of the world, let's say East Africa or the Red Sea or the Caribbean, start disappearing, but in the past, some people might have said, well, they're not that diverse, and most of the diversities in Southeast Asia and Australasia and Polynesia. Don't worry. It sounds as though we should be more worried about regional extinctions of coral reefs.
Absolutely. Yeah, that's I guess one of the really key aspects of this work. And so, for example, even if you look at the IUCN Red List, many species of corals, considered to have very, very large effective population sizes, so therefore unlikely to be globally extinct. And even getting into things like, subtropical reefs in southeastern Australia, places like Lord Howe Island and the Solitary Islands, these areas have been proposed as potentially being refuges because they're cooler. But what the work is showing is that these areas have extremely distinct corals. And so these are probably species endemic to this one region that, exist on only a few tiny little patches of rock in the middle of the Southwest Pacific. So far from being refuges, these are probably, a lot of the most threatened corals in the world. They exist in a small area. They've developed in real isolation. They're isolated from other areas. And they're adapted to cooler conditions. So, the scale of marine heat waves we're seeing at the moment, this is where we're likely to see extinctions. And without doing this taxonomic and phylogenomic work, these would likely represent what we call silent extinctions, where these species go extinct before we even know that they exist.
Yeah. And to quote E.O. Wilson, who sadly, of course, passed away quite recently, arguably the father of biodiversity, loved diversity, and he often said many times, the most important part of intelligent tinkering is to keep all the pieces. So if you don't even know what the pieces are, if you take the machine to pieces and you don't even know that there's actually 7 kinds of cogs that all look very different rather than one kind of cog, you'll never put the machine back together. So understanding all this diversity is incredibly important. And to be honest, it's intriguing to me as somebody, I just thought something like coral taxonomy was done and dusted 30 years ago. To know that it's in this revolutionary phase is very exciting. I mean, are there any stories of surprises or even contention, I suppose? Are we seeing whole genera emerging that we didn't think were genera before, not just species? Are we seeing people saying to argue, I mean, obviously people's interest in these type specimens, and start to fight about the taxonomy of these coral reef species?
It wouldn't be science if there wasn't disagreement between groups.
Yeah.
Look, absolutely. There's, there is whole new genera being created. There's been, there was a paper that one of our colleagues wrote called Sorting Out the Big Messidae, I think. Big Messidae being a reference to a particular taxonomic group. Again, one of these groups it was defined by particular characters, and then we found that those characters were really spread far and wide across the tree. So that character had evolved independently multiple times. So it wasn't a good one to define a particular group. So look, it's certainly there's been a lot that needs to be a lot of work to be done. A lot of the higher level stuff because by that I mean families and genera and things like that. Because those higher levels are more diverged than species are, because of the hierarchical nature. A lot of these older markers have already resolved a lot of that. Right. So, there was, for example, there was a famous paper in the early 2000s showing that Atlantic and Pacific corals that we thought were quite closely related because they look the same have actually been diverged for 30 to 40 million years. Right. So they're completely different groups. So a lot of that's been, I guess, known for a while. Yeah. But with these increasing increasingly complex genetic techniques, we can look in much more detail at the tips, which is the species, particularly these very diverse groups like staghorn corals. Yeah. So that's, I guess, been a really important part of this work. But also, I think it's important to remember that genetics has been, and phylogenomics has been a really disruptive technology. Right. In a good way, in many ways. It's allowing us to resolve all these questions that, we never would have thought of before. And this is far from unique to corals. Every group, that's one of the interesting things about speaking taxonomists. Every group every taxonomist that you speak to, their group's just been completely,
changed.
Absolutely, fundamentally altered what we thought. My old boss, when I started at the museum, John Hooper, worked on sponges, and he was the world authority on sponges. And he started doing a lot of genetic work, and he wrote this huge book, Systema Peripheral. And I remember him saying to me when I started, and we were talking about this, he's like, "Oh, well, throw that in the bin. I've got 5,000 species of sponges in my collection at the museum. 3,000 of them are undescribed." And so I've got a lot of work to do. Yes. Yeah, it is, and it's just so common, but it's very exciting at the same time.
And before we started, Peter, you were showing me a little machine that actually does do some of this work and getting the DNA. Can you tell us a bit about? I mean, I did biochemistry before most people here were born 40 years ago, and just getting a few base pairs could take a whole day, let alone 600 million. Where are we up to in this space in terms of the technology?
So currently what the sort of the more like modern way of doing genetics is you'll extract your DNA, you'll prepare that DNA in a certain way, and you might send it off to a sequencing facility, and a couple of months later they'll send you back the raw data, which is the genetic base pairs. But what is happening now is there's these new technologies coming out, and one piece of technology in particular is from a company in England called Oxford Nanopore. And what they have are these little devices. Okay, this is called a mini ion, and what it is a device that you plug into your computer. You put all your chemistry and your DNA into the device, and it'll actually sequence that DNA in real time and give you an output, and you can see the amount of DNA that comes out, and it can be millions of base pairs.
Yeah. I mean, it's amazing how what this is going to do for so many aspects of human society, let alone coral reefs. But exactly. We've had a really good discussion now about the importance of sorting out who is who in the zoo, effectively, in the coral reef world. These hard corals form the basis of the Great Barrier Reef. People often say it's the only organism you can see from space. Actually, well, it's a reef, it's not really an organism, but anyway, I always find that analogy very scary. But it's such a huge asset to Australia. We're going to start segueing across now to talk a little bit more about what can we do to deal with issues like climate change and sediments and nutrients. We know the coral reefs are under enormous threat. We've now learned a lot about how ridiculously diverse they are, and it would be sad to lose these systems even before we'd actually unravelled all this diversity. So I will bring you two guys back later. Thanks a lot, Tom and Peter. We're going to now cross across to Madeline Van Oppen, and Madeline is both at the Australian Institute of Marine Science and also at the University of Melbourne. And Madeline's expertise is trying to understand how we can make our corals more resistant to heat stress and climate change. Welcome, Madeline.
Thank you, Hugh. It's great to be here.
Yeah, can you, I forgot to do at the beginning is remind people that corals aren't just one type of thing. They're actually a slightly more complicated organism. And if I try and explain that, I'm going to mess it up and they'll sack me as chief scientist. Can you tell us exactly how a coral works and how the algae and the zooxanthellae all fit together?
Yeah, that's right. So corals are not just animals, they're actually animals and plants, where the plants are the tiny microalgae that live inside the cells of the corals. And so the coral host is the animal, and then we have the algae. And the algae use the sunlight to fix carbon dioxide and turn that into sugars. And a lot of the sugars are actually translocated from the microalgae to the coral host tissues. So the host relies on these microalgae for a large proportion of its nutrition.
Right, and of course what you see is what the animal makes as a shell effectively. I mean,
that's right. Shell, excuse me. Yes, so the coral animal then deposits a skeleton, a calcium carbonate skeleton, and that actually forms the three-dimensional structure of the coral reef. That is basically the real estate of the coral reef, and it's home to many other reef-dwelling organisms such as fish and other invertebrates.
So what your research is involved in, obviously, we know, and tell me if I get this wrong, that the coral bleaching occurs largely because under heat stress, the algae get expelled out of the animal, and therefore, I mean, they've lost their primary source of energy, and then they're sitting there and they will slowly also die unless they can recruit new algae. But how can we stop that? And you're working on ways and mechanisms, evolutionary mechanisms, to try and minimise the damage caused by heat stress. Can you sort of explain why the coral bleaching happens, and maybe some of what you're doing to fix it?
Yes, yeah. No, that's absolutely right what you said, Hugh. So it's believed that bleaching tends to occur during summer heat waves, and those summer heat waves tend to occur with very still weather. So the corals are not only exposed to a high seawater temperature, but they also see a lot of light. And the combination of the high light and temperature is believed to damage the photosynthetic apparatus of these microalgae, and that results in the overproduction of toxic molecules called reactive oxygen species. So they cause oxidative damage in the algal symbionts themselves, but also some of these toxic molecules leak to the coral tissues. And it's believed that these then trigger an immune-like response that results in the loss of these microalgae from the coral tissues.
So when you say immune-like response, the animal expels, or its immune system kills or expels the algae?
Yeah, okay. That's exactly right. And yeah, there's some other hypotheses now where, it's not quite clear what is the primary driver of this loss, whether it's actually these toxic molecules or whether that's a secondary response, and whether it's first that exchange of nutrients between coral animal and microalgal symbionts that actually drive the bleaching. Because once the microalgae is not performing as a good symbiont, meaning that it doesn't give the host enough nutrition, the coral might get rid of it. So it's definitely bleaching is related to these reactive oxygen species, but also to a breakdown of that nutritional relationship between host and symbiont.
Yeah. So there's a lot going on in a tiny space, and it is obviously. The one thing I do know is what is a mutualist or a symbiont, something that two species living together, helping each other, that's always a tenuous relationship, isn't it? Because at any point in time, any individual can be thinking they're not getting enough out of it, or they're feeling as though they're being a host, and that what's in them is a parasite, and they may just have had enough of this relationship.
That's right, and the mutualist, the microalgae can turn into a parasite, and that's when the coral gets rid of it.
Yeah. Very interesting. Can you tell us a bit about your research and in terms of assisted evolution and how this might actually help us solve some of these terrible problems?
Yeah, so assisted evolution or human-assisted evolution refers to bioengineering approaches that speed up the rate of adaptation of an organism to improve certain traits. And it's important to note that this doesn't include gene technologies to develop genetically modified organisms or GMOs. We are only sort of accelerating processes that already occur in nature, and we're speeding these up. And for corals, assisted evolution is aimed at increasing tolerance to warmer temperatures. Now, climate warming and associated summer heat waves are currently the biggest threat to the persistence of coral reefs. And there is concern that the rate of climate change is outpacing the speed by which corals can adapt naturally. And this is where assisted evolution is relevant, because it can speed up that rate of adaptation.
So is it a bit like how we turned a wild boar into a pig that we've bred to be different sizes and different kinds of meat? Is the assisted evolution is really just a kind of animal breeding? Well, are you breeding the algae, or are you breeding the animal, or are you breeding them both at the same time?
Yeah. We're breeding both, and you're right. It is very similar. We actually we are employing methods that have been developed in other biological systems, mostly agriculture, and most of the food that we eat has been selectively bred over hundreds, if not thousands, of years. So we're using very similar methods. So in my group, we mainly use three methods. The first one is that we take the microalgae out of the coral and then culture them in the lab and heat evolve them in the lab to increase their heat tolerance. And in the image, you can actually see some of these algal cultures that we grow in the lab. And once we have increased their heat tolerance, we then reintroduce them back into coral. And we have demonstrated that in some instances that actually does increase the heat tolerance of the coral itself as well, which is really exciting. And then the second approach is where we cross corals with a colony from a warmer region, a colony of the same species, to breed heat tolerance alleles that are encoded in the genome of the coral animal into that population of corals that lives in a cooler area, so we can prepare them for further warming. Or we cross different coral species, and that increases genetic diversity and adaptive potential. And then the third approach is not about the coral animal nor the microalgae, but it's about developing a bacterial probiotic. Very similar to we take probiotics to improve the health of our gut microbiome. Corals also live with a wide diversity of bacteria. And we are exploring whether we can manipulate these bacterial communities and in that way help the coral to cope better with climate warming.
That's a lot of different stuff. I mean, it's a lot of different ways of assisting the process, which is intriguing.
Yes.
And similar, very similar to normal animal breeding. The first one you talked about is, let's take a population, let's stress them, see who survives, and breed them for those circumstances, which is what we might have done for cattle or pigs or wheat or barley for, as you say, thousands of years. But the second one's a lot more, complicated, because you're dealing with the interactions. And the third one, you're dealing with the whole multiple species. You're evolving entire communities of organisms, aren't you?
Yes, and you hit the nail on the head where you said the interactions among the organisms are very important. Like the coral animal doesn't respond to the environment, in isolation. It interacts at the same time with the microalgal symbionts, with the bacterial symbionts, and there's a host of other microorganisms that live in and on the coral. There are fungi, there are viruses, and we know very little about these other microorganisms and what their roles might be within the sort of functioning of the coral.
Yeah, but the parallel with human medicine is also intriguing. I mean, 50 years ago, if something was wrong with a human being, we had a very basic set of tools. We have antibiotics and we had surgery, and you chop things off, and we had, bits and parts of a human being. But now, of course, we're realising so much of human health is a function of all the other things that live in a human being. The tens of thousands, I always get very distressed to know that there's tens of thousands of species living in me. And the health of those things is telling us a lot about us as individuals. And you're saying, of course, this is the same for corals.
It's exactly the same for corals, absolutely right.
Yeah, so it becomes quite a complicated puzzle. Do you get much resistance? Or, I mean, I know some of your work has been spectacularly successful in the scientific world, and obviously much of what you want to do is transfer this to helping give our coral reef systems more resilience. Is the support always unanimously positive, or some people are a little bit wary about this?
People are a bit wary at times, particularly in the beginning. We were misunderstood a little bit. People thought that we were proposing assisted evolution to replace action on climate change, and that is absolutely not the case. Assisted evolution, I see it as a way to buy time for coral reefs until we, and I mean the global we, the world actually addresses climate warming and we bring that to a halt. Yeah. Because if we don't deal with climate warming, assisted evolution can help us a little bit, but coral reefs will eventually still die. So that was sort of one misunderstanding that I think people now see that that's not what we intended to achieve. So we're only trying to buy time. Also people sometimes ask me the question, do you feel that you're playing God by, tweaking the genetics of these corals, and how do you feel? What are the ethics of assisted evolution? And again, we're not. We always initially focus with the genetic diversity that is present in a certain region. So we would always first work, say, with the Central Great Barrier Reef. If we're trying to restore a coral reef there, we would first look at the genetic diversity and the microorganisms that are present there before we go further away. So that reduces the risks associated with some of these interventions. And we're also very aware of the fact that any of these genetic manipulations, particularly selective breeding, but even just culturing coral larvae or even fragmenting corals in the lab can lead to a loss of genetic diversity, and that means a loss of adaptive capacity. And so we really need to keep that in mind, that when we manipulate these corals, that we're not losing genetic diversity.
Yeah, but in a sense, you're not doing a lot more different than what people have done with plant breeding for centuries. And of course, they have created species often, or crops, that have very little genetic diversity, and we recognise the vulnerability of our wheat and barley crops because they're not wild type, they haven't got the resilience. And so you're actually already addressing this from the get-go and saying, while we're trying to push them in a direction, assist their evolution, you want to maintain all that wild type diversity in these systems.
That's exactly right, because there's one difference. If we will implement it, if we are to implement this is going into a natural environment and not on a field like a crop. So that is quite different. Yes, so we definitely keeping an eye on that.
Yeah. So what are the calls that we heard, and we'll come back to the other two in a minute, and even the phylogenetic tree of these corals is uncertain, so presumably there's a bit of hybridisation that goes on naturally. Do you feel as though you're being sounds as though relatively cautious? Are there calls to say start looking at options of bringing in a closely related species from the Red Sea? Now I'm told the Red Sea has a fairly heat-resistant coral reef system, and a lot of species that is going to be a big distance. Is that on the table for what you're doing?
I'd say not in the first instance. I think from genomic methods, we can learn from the mechanisms that these corals have evolved, and we can look for genomic signatures in these corals and see whether they are present on, say, the Great Barrier Reef. And if they are, we can then harness that information in our assisted evolution approaches. In that manner, I think we will use it. But in terms of translocating corals from, say, the Red Sea to the Great Barrier Reef, I think we're not ready for that yet. I think we haven't explored the less risky options, the closer to home options. And also, it's not to say that these corals would thrive, because there's other environmental conditions that are very different in the Red Sea. For example, salinity levels tend to be quite high, much higher than here. So it's not a given that they would thrive here on the Great Barrier Reef. So who knows? Sometime in the future, perhaps, but not in the very recent, very near future.
Thank you, Madeline. And you'll stay there, and then we're going to bring back Peter and Tom and have a more general discussion. They've all been listening in. We've all heard what each other said about the conservation issues around this science. So we're talked a lot about the diversity and how important that is for conservation. If you don't know what you've got, it's very, very hard to conserve it. But we've also talked about assisted evolution, which is trying to get corals to be more resistant to climate change and heat and stuff. Maybe going back to Tom, from a taxonomy perspective, what do you think if some of Madeline's corals, which have been rapidly evolved, they haven't been genetically altered like a GMO, but they've been rapidly evolved in the lab, start spreading through the central Great Barrier Reef, does that cause any havoc with your taxonomy?
Yeah, who knows? Yeah, look, it's an interesting question. I guess you always see signatures going back and doing biogeography or taxonomy. Essentially what we're doing is looking for signatures of divergence or introductions. When did particular species or lineages get into particular areas? We know that the corals on the Great Barrier Reef, they haven't always been on the Great Barrier Reef. Acropora looks like, they started 60 million years ago over in the Red Sea. Or somewhere in that part of the world anyway, North Africa, the Red Sea, there wasn't the Red Sea back then. But you're often seeing, this is the constant history of life is species introductions and species extinctions and biological invasions. I guess it's a really complex question, that one. I'm not quite sure how to...
Life's always changing. I suppose sometimes I think the public, I've always thought that sorting out the phylogenetic tree was something you did, and then it was sorted out. But that was never true, was it? Because species continue to evolve, and the fact that some of Madeline's work may speed up some of their evolution, does that really matter? The fact there is no such thing as taxonomy is done and dusted. The phylogenetic tree will never move again. Even people talk about dogs, and I say dogs are wolves, and nobody's even sorted all that out. What are dingoes? Are they wolves or are they dogs? And are they all just one big species? So these debates rage, and they're changing very rapidly before our eyes.
And species don't have they have a clear definition in some senses, but there's a process of speciation where at some point you've got one species and everyone would unanimously agree that that's one species, and then at another point in time or in space you'll have two, and then there's a whole bunch of processes that lead to speciation, but they don't all occur at the same time. Sometimes in some speciation events, some processes will occur before other ones. So, at what point do you draw that line? And, because we're humans, we like to classify things into boxes, and taxonomy is hierarchical, and that's the only way you can make sense of nature, but it doesn't work that way, and everyone acknowledges that.
And we'd like it to stay fixed, I think, as well. I think we're inherently a little bit conservative. This is a left field question. I don't know if anybody can answer this, but maybe Peter. What do we know about the taxonomy and the genetics of the algae? So we've just been talking about the taxonomy of the animal. Do we have people studying the algae? Are there is there 10 algae species for every animal, or is it one algae species for 10 coral animals?
I know they had a massive revision recently.
Yeah, they did. So the main Symbiodinium, which was one of the main Zooanthellae, they have now several genera that used to just be called clade A, clade B, clade C. But now they've actually put names on them, and so that's come from genetic work where we've been able to delineate them. And so for as much we know about the corals, we know significantly less about their Zooanthellae. And that's the other black box that we also need to understand as well.
Does this make your life interesting or difficult, Madeline?
No, interesting. And I was going to say a very similar process is happening where additional species are being discovered that weren't known before, or we didn't realize. And of course, the morphology is even more limited in the algae than it is in the coral. So you have very little to go on morphologically. So it's all done by genetics and genomics. And yeah, the same thing. We are revealing additional species and species have been giving names, and it's very important. And these algae, they don't actually disperse very easily either. So it's possible that there are even more species of microalgae than there are of coral. And don't forget, only a few decades ago we thought it was all a single species, and all corals harbored the same microalgal species. So it's been an amazing change in terms of the taxonomy of the algal symbionts.
So that's quite the revolution.
I'd also like to say, that there's as many species concepts as there is species, nearly.
That's right.
Like even scientists can't agree on how you define a species. There's several different species concepts or definitions. And I think as well, it's interesting to note that like species themselves are a construct that we make. Nature may not care about species themselves.
Yeah.
Just the job they do.
And to say something that I actually know something about, which is birds, they're talking about, when I grew up, there were 10,000 bird species in the world. A few years ago, they added 700. And they're talking about there being 17,000 species of bird in the world when they finished all the taxonomy. So I thought birds was something we knew about. Hey guys, what's going on? We're doubling. It means my task of seeing every bird in the world becomes almost impossible now, so I've given up. But yeah, there's a lot of change in taxonomy. Do you feel as though some of this taxonomic change actually, I mean, we talked about keeping all the pieces, and if go back to E.O. Wilson and the conservation and say, well, we need to keep all the species, people sort of think, yeah, we should keep every kind of species of kangaroo, and there's 3 species of wombat, we should keep all them. If there's 150 corals, maybe we've decided on the Great Barrier Reef, we should keep all the pieces, but you're saying we don't even know what the pieces are, and even the definition of what is a piece, we're going to fight about. Madeline?
We may not be able to keep all the species or the pieces anyway. So then we need to focus on, we need to keep at least enough so we can have a reef ecosystem that supports other species that, as we call it, is functional. And the question is, of course, who are those species that we actually need to maintain? And with hybridization, of course, it's going to confuse the taxonomy, but we need to keep in mind at least it will preserve the genetic diversity that is present. So whether that genetic diversity is in 2 species or in the one hybrid species, at least you maintain that genetic diversity that was present on the reef.
So you've sort of raised 2 issues there, which I think is foreigns a lot of people in conservation. The focus is always, the first thing is how many species are we losing? That's the first thing people talk about. But you're actually saying it may be more about functions. Are we keeping all the functions, or are we keeping all the evolutionary diversity, all the genetic diversity? And maybe that's more important. Of course, the public don't talk about that.
In an ideal world, we would want to keep all the species, but I think if we're realistic about conservation and restoration, that's probably not feasible. And I think then we have to make choices of what is important in terms of maintaining a functional reef.
I guess one of the other important things as well with species concepts, that's absolutely right. But again, going back to the idea that, people need to, like to put species in boxes. If we think about how we go about conservation, you look at, the Queensland, it's the threatened species list, or, the IUCN red list. If we're going to say, well, you want to build a port over there, you have to say, is there any threatened species in there? So while it's, in some ways, it can be an esoteric argument to say, well, this is 1 or 2 species. Birds are actually been a classic example and test case of this a lot, where you say, well, this species is widespread. Oh, no, actually it's 3 species and they're not just separate populations. So therefore this one's now critically endangered. Therefore you've got to implement a management plan or whatever. So I guess that's a really important thing. There's scientific questions in terms of, how much evolutionary diversity are we protecting or whatever, but they're not necessarily the same or thought about in the same way as the practical implications, which is that a manager says, well, how many threatened species live here, or, how many of the species that live here are threatened or whatever. So there's sort of, I guess, both questions, which is, I guess, again, there's not an easy answer to that, but
And legislation, I suppose, which is the politics of it, and also the common public's opinions of things, doesn't necessarily deal well with a lot of the complexity you've just hurt my brain about dealing with coral conservation. How do you deal with it when it is so complicated? And there will be scientists I know who will focus more on function, some focus more on genetic diversity. You know, how many different kinds of a cropper do you need to keep all those lovely fish, which is probably what most people see. So this is quite a challenge for us, isn't it? Because there is a translation challenge, I think. Because if we just keep saying the species are not well defined and we need more function, what does that mean for the headline legislation in this space, which is the EPBC Act, the Environmental Protection and Biodiversity Conservation Act, which is all based on species, and a little bit on ecosystems, never talks about function and never talks about genetic diversity. Sorry, that's just a show-stopping complexity thing.
Absolutely right. Yeah.
Let me.
I think we all agree.
That's right.
That's why you're the chief scientist.
That's right. Yeah, not my. Yes, maybe it is my problem. Maybe I can end with one last question for all of you, just a little bit off the table. I mean, you live and breathe coral reefs, and you're passionate about them, and everybody in this country understands that the Great Barrier Reef, corals, the fish diversity, and they don't even see all this other diversity that you're talking about, Madeline, bacteria and symbionts. But we all have worked out that this is pretty special, and it's special for our Indigenous heritage, our cultural heritage. It's essential for the prosperity of the nation, really. I mean, tourism is our third biggest industry, but they're providing fisheries services, cultural services, protection against storm surges and cyclones and hurricanes, all these things. If we lose these ecosystems, it's a disaster. What do you? I mean, I'll go to Madeline first, and then Peter and Tom. You know, are you optimistic about the future of the Great Barrier Reef and the corals that you love?
Very concerned about the future of the Great Barrier Reef and coral reefs around the world, particularly because we haven't been doing very well in terms of reducing greenhouse gases in the atmosphere on a worldwide scale. And so we're heading towards significant further warming. And so I'm very concerned about that. I think we're doing great in the development of innovative coral reef restoration tools that will help to some extent. But again, if we can't keep the coral stock that we use for restoration alive because it's too warm in summer, then we have a problem. So yeah, I like to be positive, but I'm also very concerned.
Peter.
So I study things on geological timescales. You know, I'm looking at patterns from 60 million years ago, and you know, I've spent a lot of time looking at like past extinction events, and you know, like reefs have been around for 400 million years. You know, there's been some sort of shallow water reef type habitat for that time, and what dominates that habitat has definitely changed. And what we see today is dominated by hard corals, and that system is very beneficial to us. So reefs are going to go on, but it's whether we can adapt to that changing reef environment as well. And so the Great Barrier Reef has already changed. It's not the same Great Barrier Reef it was 20 years ago. And so the species composition has changed. Every mass bleaching event changes the composition of the reef and the dynamics of it. So I think I'm very concerned about how it's changing, but more from the fact that how are we going to change with it, and are we going to implement what needs to be done to slow that change?
Tom.
Yeah, thanks. I guess, I mean, the first important thing, I think it's unanimous amongst the coral reef science community, just to reiterate what Madeline was saying, that, you know, carbon emissions need to be brought under control. And that's not just for coral reefs, that's for all sorts of ecosystems and whatever. That's something that, you know, we like to argue with each other about various aspects of, you know, what we should do or what species are. But, you know, that's really a unanimous, viewpoint amongst scientists. But specifically, I guess, you know, I'm a bit like Pete. I have a long-term, you know, perspective as a, you know, paleo, ecology background. And so, yeah, reefs have come and gone over huge periods of time. These are cropper-dominated reefs that we see now. They're quite anomalous in the history of even, you know, modern coral reefs have really been around for about 50, 60 million years. And the reefs that we see now and the species that we see dominating them are that's really only happened recently. And we can see that from the fossil record, and we can also see that from the genetics. So that's, interesting. Well, what were reefs like before that? How did corals live? How did they function? And if we can work that out, what does that mean for us? And then going back to what Pete said before, reefs have been cradles of biodiversity for hundreds of millions of years. But we've also published a paper that just came out very recently looking at huge reef systems from the Devonians about 385 million years ago, the biggest reefs in Earth history. And those reefs collapsed, coinciding in the late Devonian mass extinction with spikes in temperature. And we really didn't see coral-dominated reefs like that for, 150 million years. So they can have really long-term consequences as well. So I think they're interesting questions, and it's a difficult question to answer, that there's, there's change, absolutely, and things have always changed, but there's also implications for those changes. So trying to understand what those are and how that affects people and ecosystems and evolutionary diversity is really important.
Thank you all very much. Let me thank you, Tom, Peter and Madeline. I learned an enormous amount. I mean, I've come, I thought I knew a little bit about marine ecosystems, but I need to do another undergraduate degree or 3, or spend some time in the museum or at AIMS working out what's really going on. It's wonderful. Also the fact that even though you've learnt so much and done so much, we are the world's centre of coral reef studies, there's still so much to sort out. And so many things to learn in this system, so we can help to manage it. To your point, I will be taking the train home, so I'm doing my bit. I drive once a month, and I post regularly on all the things that every person can do about climate change. Climate change is the big threat to both humanity and these ecosystems they love, and particularly coral reefs, which are so important. Let me thank you enormously for your participation. I'd also like to thank the people here, the team here from Griffith who've run this, the museum, the World Science Festival Brisbane. I've enjoyed this a lot, and I hope you enjoy watching it too.